so yeah, we'll switch to another topic on enteroviruses. According to the name, you can see that this is a big group of viruses which can infect in the digestive tract. Entero means a digestive tract. Enteroviruses, and this is a big name, a big group. It includes a lot of types of viruses, and we'll focus some of them. This is a genre of picoviruses, pico RNA viruses. In this name, you can see in this name, pico RNA viruses. Here, pico means small. Pico means small. RNA in the middle means that these are the RNA viruses. So enteroviruses are pico RNA virus, small RNA viruses that include enterovirus and also rhinovirus. And today we'll focus on enterovirus, especially we'll focus on polio, because polio is a major threat in some countries, not every country. In some countries, WHO set the goal that we will eliminate polio virus in recent years, but it seems that due to a lot of reasons, we cannot achieve the goal on time. In China, polio virus is almost gone, and I think ever since 1990s, that's more than 20 years ago, 20 or 30 years ago, there was no polio cases anymore due to the wild virus infection. So that's because of the help of vaccines. And later we'll talk about the vaccines. And right now there are only 4 countries with polio viruses, including I think Pakistan, Afghanistan, and another 2 countries, maybe 2 countries in Africa somewhere. And because the vaccines are not well applied in those countries. Here are the categories of enteroviruses, and we'll focus on poliovirus first, and we'll also talk about the Coxsackie virus if we have time. The properties of enteroviruses. Here are some properties here. They are small, as you can see. As you can see, the size of enteroviruses is about 28 to 30 nanometer. That's much smaller than flu virus. Flu virus is about 100 nanometer. Their genome is a single-stranded RNA, positive sense RNA. So there is only one RNA genome, one strand, single-stranded RNA and linear. They can produce proteins, including the VP1 to VP4. They don't have envelope, and their replication is in the cytoplasm. That's not surprise because this is RNA virus. After encoding, they stay in the nucleus, and they stay in the cytoplasm. They don't have to enter into the nucleus. Transmission of enterovirus, they are setting infection in the digestive tract. It's a fecal-oral route through contamination, through the ingestion via contaminated food and water. So this is the transmission. Or contact with infected hands, or sometimes inhalation of infectious aerosols. Different transmission route, either ingestion, direct contact, or respiration of infectious aerosols, continuum viral particle. or fecal-oral route. Here are the different enteroviruses. There are poliovirus and also echovirus, Hep A, that's hepatitis A virus, also belongs to this group. And different viruses can enter the host in different ways. Some through the entry of our aerosol or ingestion, some through like other ways. And they can cause different disease. In this table, it shows us several important viruses, including polio, COX-A, COX-B, and also echo. And we'll focus on polio. Polio is, long time ago, it was a big threat in the history in all over the world. In 1940s, 1950s, polio infection was a major threat in America, and some people, even adults, got infected and got disableized. For example, the famous President Roosevelt, he was a victim of polio infection. Yes, there are rumors going around in some countries. The people there believe that vaccines, not only polio vaccines, a lot of vaccines can cause damage to the reproductive system. And they said that the rumor said that vaccines are from Western countries, from enemy countries, and those countries want to use vaccine to kill those people so that they won't have babies. That's the problem. When WHO want to use flu vaccine in some countries, there was also rumor about vaccines. They said that we cannot use vaccines. This rumor is propagated by especially those religious groups, the dramatical religious groups, and finally WHO find a way to solve the problem, because the people there don't trust WHO, don't trust the Western countries. They only trust those countries with the same religion. So they finally find a better way. They allow those countries who share the same religion to produce the vaccine. So they asked Indonesia to produce vaccine for those countries. And that is better. And they think that those vaccines are safe. And because those countries are sharing the same religion, and that WHO can spread the flu vaccine there and can protect those people. Okay, so we'll focus on polio. Polio infection can cause either infection without symptoms, or sometimes they can cause meningitis, that's influence in the nervous system, and also they can cause paralysis. This paralysis can be on legs, but can also influence other parts, other muscles in the body. Immunity against those viruses, antibody is a major protective immune response to enteroviruses. So we can induce the system to produce antibodies. In those antibodies, secretory antibodies can prevent the initial establishment of infection in the airway or in the gastrointestinal tract. And serum antibody in the blood, it can prevent viremia spread to the target tissue and therefore stop the disease. Cell-mediated immunity is not usually involved in protection, but may play a role in pathogenesis. This is an immune response to enteroviruses. Then we'll focus on the major part in this section. We'll focus on the poliovirus. This is the important characteristics of poliovirus. The picture on the left are the virus under electron microscope. You can see they are pretty small, and this is a schematic structure of poliovirus. There is a single-stranded RNA genome, positive genome, and a protein shell, the capsid. On the capsid, there are several important proteins, including VP1, VP2, VP3, and VP4, forming this capsid. Source of infection. Polio. It's dangerous because poliovirus can be transmitted from person to person, like from either some apparent patient showing symptoms or from someone who are not showing symptoms. We call it subclinical patients. Both of them can be the source of virus. The port of entry is from a mouse. So it's from the ingestion of the virus. Virus is regularly present in the throat and in the stools before the onset of illness. So before there is any symptoms, the virus is already in the throat or in the stools. So that is why it's not easy to prevent the poliovirus if there is no vaccine. Poliovirus can spread along axons of peripheral nerves to the central nervous system, that including spinal cord and brain. So it can cause meningitis in the brain. This is poliovirus. Clinical syndromes. The incubation time for poliovirus is 7 to 14 days. That's pretty long. And during this incubation time, there is no symptoms. The reason we call it incubation time because that's before the onset of the disease, before the onset of the illness. It's not detectable by symptoms. But before the showing up of the symptoms, it's already infectious. It's already, it can spread the virus to other persons. Of all the infected persons, inapparent infection is the majority part. It takes 90%. And, without any noticeable symptom, without noticeable disease, that person is a source of infection. It can cause different disease if there is some episode of disease, including fever, malaise, headache, and for a very small portion of it, it can cause severe disease, including like non-paralytic poliomyelitis. We call it aseptic meningitis. So there's no bacteria, and a viral infection can cause meningitis. But this is in the nervous system. It doesn't cause paralytic symptoms. While for the paralytic symptoms, it's noticeable, and you can tell which one is infected by poliovirus. It causes paralytic poliomyelitis. That's a flaccid paralysis. The major illness we can identify. When I was a little kid, in when I was in primary school and in junior high school, I can see a lot of people on the street who are flaccid paralysis because they were victims of poliovirus. And when I was in junior high, we have several teachers, and 2 of them are victims of polio infection. And they have flaccid paralysis in their legs. They cannot walk well. They could not walk well. That's that's a long time ago in junior high. But now those polio infection cases are much rare in China. But flaccid paralysis in legs is not the severe disease it can cause. Sometimes polio infection can cause trouble in the muscle controlling your respiration, in the muscle controlling your breath. If that happens, it's life-threatening. It can cause infection in the nerve system, and the muscle involved in respiration is weak. The patient they don't have enough strength to take in oxygen. That's very dangerous and very critical. So during that time, a lot of patients need to rely on iron lung to survive. The iron lung is a chamber, so that the patient need to lie in the chamber and with their head outside of the lung, and the chamber can help the patient to breathe, so that they can get enough oxygen. And even right now, there are some elder persons in America. They were victims of polio infection when they were kids or when they were young. They are still relying on the iron lung to survive. Let me play a couple of videos on the iron lung. Maybe this one first. Sorry. Let me turn on the audio. Just now, can you hear the audio? No? Let's try now. How about now? Great. Pay attention to the structure of the iron lung.
Hi, I'm Cassie Nesper from the Rose Malnick Medical Museum at Youngstown State University. Today we're taking an in-depth look at the iron lung. Let me get out of here so I can show you around. Iron lungs usually refer to respirators that encase your entire body. They can be used in any kind of illness that impairs your breathing, such as paralysis of the diaphragm or severe cases of polio. They work by varying the air pressure inside the chamber. When the pressure becomes less than that inside your lungs, you inhale and air expands to fill your lungs. When the pressure is more than that inside your lungs, you exhale. Devices similar to the iron lung have been around since the 1830s. Philip Drinker and Dr. Lewis Shaw were the first to use the term iron lung to describe their machine in 1928. It was used widely in the polio epidemic that year. John Emerson, who ran a medical machine shop nearby, also saw their model. He made some improvements, including ports on the side where nurses could access the patient for care, and a bed that slid in and out. His first machine is now part of the Smithsonian Institution at the National Museum of American History. During the polio epidemics of the 1940s and 50s, American hospitals had wards full of polio patients and iron lungs. So let's turn this thing on. The switch for the motor is on the front, along with a switch for the light inside the chamber. At the bottom is a knob that controls the respiration rate of the machine. It can go between 12 and 20 breaths per minute. Along the top is a gauge that measures the pressure inside the chamber. Next to it would have been an alarm where a light would go off if the pressure inside the chamber goes below a certain level. In case of electrical failure, there's a handle at the rear of the machine for manual operation. Along the side are ports for nurses to access the patient and windows. At the head of the machine is the collar that fits snugly around the patient's neck. This would have been made out of a sponge material or a plastic. There are also knobs to adjust the bed position inside, and a mirror so the patient can see what's going on in the world around them. This could also be a book rest, although somebody else would have to turn the pages. If a patient were in the iron lung for polio, they would be in there for about 2 to 3 weeks until they got well enough to breathe again on their own. So there you have it, the iron lung. Join us next time for a tour of the wooden X-ray machine.
Okay, so that's the iron lung. Have you ever heard of this iron lung before our lecture? No? Okay. So that means online course is also useful. You learn something, right? Okay. Okay. I have another video. Maybe we can play it. Do you have other courses after this one? I don't think so. Okay, so I'll play another video for you guys. And enjoy our lecture time. This is a real patient using the iron lung.
Yeah.
I was working in the job, had quit college to get married, and got sick my third day at work. And felt like I had the worst case of flu in the world, the bursting head and the whole thing. Everything was extremely sensitive. And I became weaker the next day. Couldn't walk then. The third day I couldn't breathe right, so rushed to the hospital. Doctors thought it was encephalitis at first, because this was 1956, and polio was on its way out. Most of the bad cases had been in the early 50s. But they realized they pulled an old iron lung out of the basement. This was St. Luke's Hospital. And so I was in that for 3 months.
Probably just a piece of him under there.
Probably 20 years after I had it was when I started having the breathing problems. So I went back to the hospital, and at that time they decided I needed the lung to sleep with. Now they're believing it was post-polio. You don't get the disease again, but the little nerves that you were operating on get weaker. This is a real character blow for me. I was always very independent, and learning to depend is hard. The most of the work in your breathing is done by your diaphragm, and so it gets very tired after all day, after working with fewer muscles. And the lung just takes over the breathing for me when I'm in it. It's a relief to get in, it's a relief to get out. It's like, you're ready for it at night, tired. Just go right to sleep. Then in the morning, you're ready to be out. The fact that it's available and the statistics are so good for the prevention, it's just a wise thing to do, I believe. People are afraid that they hear stories of cases where people got polio anyway or some other dreadful thing, but it's so rare. It's worth taking that little bitty risk is to let it go rampant again and be epidemic, which could happen.
Okay, so that's a sad story, and it happened a long time ago. In 1940s, 1950s, in America, especially in America, different groups of scientists tried to fight against polio infection and try to make vaccines. And there are some interesting stories about the vaccine development. And finally, some vaccines are available, and some accident really happened during the process to make vaccines. And for now, we look at those stories as experience or lessons, but when it happened, a lot of people were influenced. So this is a sad story. And now another story about the iron lung is that the people still using the iron lung are only several of them because they are getting old. And the market for iron lung is very small. So the manufacturer stopped making iron lung because there is no market. So those people are facing a problem of parts, and they have to use the iron lung every day. So some parts of the iron lung need to be replaced. The problem is that the manufacturer is gone, and they cannot get new parts for replacement. So that's the problem they are facing. Okay, these are the victims of paralytic polio, the kids who are infected by the polio virus. And this problem can stay with them lifelong, and they cannot walk well, and they don't have strength in either one of the legs or the two legs. So this is paralytic polio victims. For immunity against polio virus, secretory IgA and neutralizing antibodies, including IgG, IgA, and IgM, can persist for lifespan in the patient. So the patient can produce antibodies. And for prevention, there are two types of vaccines. One is the oral polio vaccine. This is short for OPV, oral polio vaccine, OPV. It includes attenuated viruses, attenuated live virus. If you think a virus is a live organism, it's attenuated. And we can give the kids attenuated live virus through oral. So you make a viral suspension in serum and you drop it in the mouth of the babies so that they will ingest the attenuated live virus, which can stimulate the immune system. Another type of vaccine is IPV. Okay, cool. OPV for children. Yeah, they were made by Salk and Sabin. And this IPV is a new type of vaccine, which contains inactivated polio vaccine. So this is inactivated poliovirus. And the administration route is injection. So you inject this IPV under the skin, which can also turn on the immune system. Compared to OPV, the activity of IPV is lower. So right now, in some countries, like in America, the kids get IPV, but in China, previously the kids get OPV regularly. But for OPV virus, OPV vaccine, there is some problem. For very rare cases, OPV can cause infection. For example, if the kid is so special, the kid's immune system is too weak. And for those cases, the OPV vaccine itself can cause infection because it's attenuated live virus. It can cause infection in very small portion of the whole population, maybe 1 out of a million. 1 out of a million kids, it can cause infection. So that's OPV. The immune response is strong. The protective effect is strong. But there is some problem of OPV. So right now, there is a strategy which can combine OPV and IPV together. So the doctors will give the kids IPV first so that it can prime the immune system. It can activate the immune system for the first time. And then they will give the kids OPV, which can stimulate the immune system better. So OPV as a booster and IPV to prime the immune system. And that is much safer than before and can also activate the immune system. So this is the what we talk about OPV. The advantage of it is that it can induce the secretory antibody response similar to that of natural infection, but it can cause and it's easy to be administered. So you just drop a serum to the mouse. The disadvantage of OPV is that sometimes it can cause vaccine-associated paralytic poliomyelitis. So that's infection by the vaccine and spread of vaccine to contacts. Unsafe administration for immunodeficient patients. Yeah, there is some immunization procedure in those countries, people can get a booster administration to set up the herd immunity to prevent the polio infection. It also happens in China. In China, every year the kids get OPV booster to eradicate the virus. But for infectious disease, it's not for any country. It's a global problem. Even in some countries, like in China, there's no infection cases for a long time. But we still need to give the kids a vaccine every year to prevent the coming back of the infection. But once there is no polio virus anymore, we can stop the vaccine. And that happens for the smallpox. Previously, we need to give everyone vaccine against smallpox. But once the smallpox is eradicated from the earth, there is no virus anymore. We can stop the vaccination. Hopefully, in recent several years, we can eradicate polio all over the world. The advantage of IPV is that first, it's safe. It's good stability during transport and in storage. It's safe for administration and no risk of the vaccine-related disease. So that's IPV. The disadvantage of IPV is that it lack of induction of local immunity. It cannot induce the mucosal immunity in the gut. And also that it needs an injection. And this procedure is more painful than the oral administration. Yeah, and as we mentioned previously, there is strategy. We can combine IPV and OPV, use IPV to prime the immune system to protect the people for the first time, and then OPV to boosting the immune response. Except for this polio, in the enterovirus, there are also Coxsackie virus and also echovirus, which can cause the disease called the hand, foot, and mouth disease. This is the hand, foot, and mouth disease, mostly caused by Coxsackie A. Can cause fever in kids less than 5 years old. Fever, malaise, sore throat, and it can cause these vesicles on tongue, hand, feet, and buttocks. So that's the typical symptoms of this hand, foot, and mouth disease. These viruses are highly infectious. Problem is that we can only give the kids supporting treatment. We don't have any specific medicine. And the other problem is that for the disease is just the symptoms of the disease. There are several different types of virus can cause the similar symptoms. We don't have vaccine against any. We don't have a vaccine against all the viruses. In several years ago in China, there was one vaccine developed against EV71, I think. That vaccine can help the kids and prevent from infection by that specific virus, but not all. It's helpful, but not very helpful, because even the kids get that EV vaccine, they should be careful to be they should be away from infected kids, and the infected kids should be isolated so that they won't transmit the virus to other kids. So this is the hand, foot, and mouth disease caused by Coxsackie virus or echo virus. Okay, and we have some video here. Do you want to watch the video here, or you want to watch the video by yourself? Okay, here. Okay, great. I think just now I made a mistake about our last lecture. I think we agreed to have a lecture tomorrow night at 7 p.m. That's because of the sorry, I'm getting old. I cannot remember the reason. Do you want to have a lecture tomorrow night or on Thursday night? It's up to you. We can combine it together. Yeah, I guess tomorrow night is good for you guys, even though I'm kind of tired because I have to work tomorrow afternoon. That's okay. Yeah, we can, yeah, okay. We'll have another lecture tomorrow night at 7 p.m. and both groups will have the lecture together, okay? Thursday is the New Year. Yeah, Thursday is New Year Eve. Yeah. That's okay for me. And yeah, okay. We'll have a lecture, the last lecture tomorrow night at 7 p.m. Everyone from both groups will join together, okay? And yeah, after the lecture, you will have 1 day more to prepare for your final exam. That's on January 6th for your final exam, okay? And yeah, I'll play some video for you. Which one first? Maybe this polio.
So the first enteric virus that we're going to talk about is the enterovirus poliovirus. And polio is a hardy, meaning that it can survive in many different environments. It's a hardy, non-enveloped, positive, single-stranded RNA virus. It has a capsid with icosahedral symmetry, and it's almost entirely eradicated thanks to vaccines, but it's still important to learn about. So what does poliovirus do? Do you know? Well, you might think of FDR paralyzed in his wheelchair, and indeed, poliovirus can cause paralysis. The last video you learned that poliovirus is an enteric virus that replicates in the gut. So how do these things go together? So let's go through it. Infection with polio starts when the virus comes in through the mouth. The viral travels down through the stomach and into the intestine where it infects epithelial cells. The fact that it goes through the stomach, again, means it has to survive in very acidic environments, and indeed it can actually survive in a pH as low as 2. So how does it infect epithelial cells? Well, it binds to a certain receptor called CD155, and then gets its RNA into the cell, and then starts making proteins and replicating. So what is CD155? Well, it's a surface receptor that's important in making structural connections that hold adjacent epithelial cells together. So it gets into the cell, and then what does it do? So inside a cell, its positive single-stranded RNA can be directly translated into protein. So it translates some protein, and then its genome replicates using its own RNA-dependent RNA polymerase. The new viral particles are assembled, which then lyse, in other words, kill the infected cell. And this lysis releases many viral particles. So some of these are going to go into the feces, and others are going to stick around in the interstitium around the cells that are infected. And so what happens to fluid and other material in the interstitium, well, it gets swept into lymphatic vessels and passes through the lymph nodes nearby. And eventually, all lymph gets dumped back into the blood. And so through this mechanism, poliovirus gets into the blood. So we call that viremia, where emia means it's in the blood. Okay, so we still haven't mentioned anything here that has anything to do with paralysis, but now we're finally there. Because once the virus is in the blood, it goes all over the body. It could theoretically infect anything. And in some very unlucky people, 1 in 1 or 200, it gets through the blood-brain barrier and into the central nervous system and starts infecting neurons. So does it infect any neurons? Well, no, not quite, because we know poliovirus mostly causes paralysis, not dementia or sensory loss or something else. So we can reason out that for some reason it likes motor neurons, not sensory neurons or other neurons in the brain. And indeed, it mostly replicates in the lower motor neurons and infects their cell bodies. So where are those? Do you remember where the lower motor neuron bodies are? And this is a lot of detail, but as you'll see, it's good to know. So the lower motor neuron cell bodies are in the anterior horn of the spinal cord. So here, this stuff is all axons, this stuff is all cell bodies, and the ones up here are the motor neurons. Now note that this is the spinal cord. These cell bodies are in the spinal cord. And what do we call inflammation of the spinal cord? Myelitis. So when poliovirus infects the spine and causes paralysis, we call it poliomyelitis. You might have heard that term before, and now you know where it comes from. So there you go. We've talked about how we go from infection in the GI tract all the way to paralysis.
Okay. I'd like to play another video. It's it's a new type of disease which is happening in recent couple of years, especially in US. And this disease, it's a polio-like illness. But I think till now, the pathogen has not been identified. It happened in US, and some kids, the children, got infected, and they developed the symptoms like polio. But the antigen detected is not the same antigen as in polio. I think people are still working on it. Let's watch this current issue. Oh, sorry. Yeah, I guess the video is damaged. Sorry. It cannot be played. Okay. Maybe the first one? The video damaged by poliovirus, yeah. If you can
Hi, I'm Sharon
prove that,
Smith. I'm a
you
pediatric
will get a
infectious
Nobel
disease
Prize.
doctor at the University of Washington. And in this video, we're going to be talking about enteroviruses, which are ubiquitous viruses and capable of producing mild and severe disease. Polio, an enterovirus that causes paralytic neurologic disease, serves as a reminder that effective public health interventions can have long-lasting effects on human health and disease. Here are the learning objectives: to outline the classification and basic viral structure of enteroviruses, discuss how these factors relate to epidemiology and spread, discuss the immune response to enteroviruses, including viral recognition by pattern recognition receptors, the antiviral immune response.
This video is relatively long, about 10 minutes.
Here's the course map linking back to enteroviruses. This is Avery. She is 4 days old, and yesterday her parents noticed that she wasn't eating as well as she had been, and this morning she started crying and they couldn't calm her down. They thought that she felt warm, and they found out she had a fever. She was brought to her pediatrician's office, who admitted her to the hospital because she was so ill. Her mother, father, both had colds, as did her older brother. When Avery had testing to find out why she was ill, her doctors found out that she had evidence of meningitis. Ultimately, she was diagnosed with enteroviral meningitis. While she was in the hospital, she had high fevers and really wasn't able to eat and was very irritable. She was there for 4 days until her fever went away and she started eating again. Ultimately, she recovered completely. Her story tells us why we care about enteroviral infection and gives a little clue about who is susceptible to this infection. So first, I'd like to talk about the basic biology of enteroviruses. Enteroviruses are small RNA viruses that belong to the picornavirus family of viruses. The word picornavirus is derived from the word pico, meaning small. Other viruses in this family include hepatitis A and rhinovirus. Humans are the only natural hosts for enterovirus, and this factor has implications for transmission and control. There are more than 100 types of enterovirus with 4 main species grouped by genetic sequences. You will hear people describing the viruses this way. For example, enterovirus D68 caused a recent outbreak of illness in the United States. When you talk to some clinicians, they may also use older categories to describe the viruses, which do not really align with genetics. These biologic categories were created by grouping viruses together that cause similar diseases. The problem with this approach is that there is significant overlap between the types of illnesses different enteroviruses can cause. This is just a heads up for you so you don't get confused in the future. Some of the most common labels that you will hear are Coxsackie viruses, echoviruses, enteroviruses, and polioviruses. Since enteroviruses are RNA viruses, their life cycle is pretty typical of RNA viruses. They attach to the cell surface via specialized receptors, for instance, CD155 for polio, and are endocytosed. The replication and assembly of the virus occurs exclusively in the cytoplasm, and the viral RNA polymerase is the key enzyme needed for viral replication. The virus is subsequently packaged and released. The release of the viruses causes cell death, and the viral particles are immediately infectious. So the clinical correlations of this are, since viral replication is relatively simple, there are not a lot of targets for therapy. In addition, there are significant similarities in the enteroviral genomes in the different groups. Since enterovirus commonly causes disease, this was one of the reasons that it was one of the earliest diagnostic PCRs to be developed. This really has changed the management of patients with common disease manifestations like meningitis. The similarities between groups may also be a limitation because some of the tests may not distinguish between enteroviruses and similar viruses like rhinovirus, which causes the common cold. I want to talk about how enteroviruses cause disease. They can infect both the GI and respiratory tract. I'll talk about each one separately for clarity. The virus is shed from the GI or respiratory tract of a patient. This virus is immediately infectious. Initial infection happens in the epithelial cells of the upper respiratory tract or oral pharynx. There is local replication of the virus within those cells. The virus then subsequently infects and then replicates in the local submucosal lymphoid tissue. If the initial infection is in the gastrointestinal tract, again, primarily the oral pharynx, there is that initial replication in the epithelial cells. New virus is shed and swallowed. The virus resists breakdown by stomach acid, which means it's acid stable, and this allows it to move into the intestines, where it subsequently infects the submucosal lymphoid tissue, specifically the Peyer's patches. Once the virus replicates in the submucosal lymphoid tissue, there is primary viremia with additional replication in the lymph nodes, spleen, liver, and bone marrow. This is followed by a secondary viremia and infection of additional organs like the central nervous system, heart, and skin. Now, let's talk a little bit about the immune response to enterovirus so we can link viral pathogenesis, the immune response, and clinical manifestations. The first line of defense against viral pathogens is the innate immune system, which recognizes viral proteins as non-self. This initial recognition of non-self is mediated by pattern recognition receptors, or PRRs. The PRRs are glycoprotein molecules that are found on many cells involved in the innate immune response. They are so named because they recognize and bind to pathogen-associated molecular patterns, or PAMs, molecular components associated with microorganisms but not found as part of a eukaryotic cell. The toll-like receptor 3, or TLR3, is the most important PRR for enterovirus. So toll-like receptors are expressed on both the cell surfaces or in endosomes where they can interact with antigens. TLR3 is in endosomes of T, B, dendritic, and NK cells, where it can interact with viral antigens after the virus is endocytosed. When a virus is recognized by TLR3, there is an activation of a series of transcription factors that result in inflammation. NF-kappa B activates transcription of a large number of proteins, including cytokines, chemokines, and adhesion molecules. Interferon response factors control the expression of interferons, which are critical to stop viral replication. Type 1 interferons increase natural killer cell activity and activate cells of the adaptive immune system, such as antigen-presenting cells, like dendritic cells, or CD4 and CD8 T cells, in part by upregulating the expression of MHC molecules. They also directly limit viral replication through disruption of viral replication processes and signaling adjacent cells to undergo apoptosis. We know that a humoral immune response is critical for the control of infection, mainly through studies in people with immunodeficiencies and through vaccine trials. There are two places antibodies are important. One at the mucosal surface, primarily secretory IgA, that, if present, can stop infection. The other is in the serum, where antibodies act to inhibit virus that is circulating. These antibodies are present if you have been exposed to the virus previously, have been immunized, or they occur during the secondary viremia that happens later in the course of the infection. So why do people get sick with enteroviruses? The first time a patient is exposed to a virus, there isn't any adaptive immune response early in the infection, and it isn't until viral replication in the submucosal lymph nodes that the innate immune system is engaged. The only way to prevent infection at this point is if secretory IgA is present from immunization or previous infection. You can see from this slide where the various parts of the immune system interact with the virus to limit spread. Control during primary viremia results in asymptomatic infection. This is due largely to an effective innate response or presence of antibodies from previous infection or immunization. If there is no control, there is additional replication and the patient develops fever during the secondary viremia. If replication is stopped there, only fever occurs. If there's no control, there's additional spread and disease occurs. So not everyone presents the same way with this illness, and some people have increased risk for disease. Young children, like Avery, are at particular risk, likely because of their impaired immune response. You can see in the slide that infants who are less than a year of age have the highest number of deaths and the largest number of infections. In addition, we know that patients who do not have antibodies will develop persistent meningitis because they cannot clear the infection without neutralizing antibodies. Finally, stem cell transplant patients with multiple defects in their immune response develop multi-organ disease, including infection of the heart, lungs, and central nervous system. In summary, enteroviruses are very, very common and are RNA viruses. Pattern recognition receptors are important for the initiation of the immune response, and the humoral immune response is critical for the control of infection. Because of the effectiveness of the immune response and the ubiquitous nature of the virus, most infections are asymptomatic. If there is no control, the secondary viremia produces fever and leads to end organ infection.
Okay, so I think that's all for today. We spent 2 hours on these 2 lectures, and we'll have a lecture tomorrow night, and see you then.